Turritopsis Dohrnii. Most people believe aging is the one biological process. Nothing escapes. Every cell degrades. Every organism declines. The direction of biological time moves in one direction only. Turritopsis Dohrnii does not move in one direction. It reverses. When this jellyfish reaches full biological maturity after it has developed, reproduced, and begun the cellular decline, every other multicellular organism follows toward death. It converts its adult cells directly back into juvenile cells and restarts the developmental cycle from the beginning. No intermediate state. No regression to a stem cell phase. The fully differentiated adult cells of one tissue type convert directly into the fully differentiated cells of a completely different type in a process called transdifferentiation that no other multicellular organism has been documented performing. The effect visible in laboratory specimens is an organism cycling through maturity and reversal indefinitely, with no documented upper limit to the number of repetitions. The cause, the specific molecular triggers that initiate transdifferentiation, has been partially mapped. The conditions that activate the reversal in turritopses are the same conditions present in aging mammalian tissue: cellular stress, damage signals, the chemical markers of age. The machinery exists. It exists in an animal the size of a fingernail. It has existed since long before any human institution began studying it. And every aging research program currently active is working on a version of the same question the jellyfish answered for itself before our species evolved to ask it. Wood frog. The wood frog freezes solid every winter, not dormant, not in torpor, frozen, heart stopped, brain activity ceased, ice crystals forming between cells, the body brittle enough to produce an audible sound when tapped. It remains that way for months. In spring, it thaws, and within hours, it is feeding and breeding. The visible result is an organism that crosses the clinical definition of death twice a year, voluntarily, without consequence. The cause is a two-stage biochemical response that activates within minutes of the first ice crystal contacting the skin. Ice nucleating proteins in the skin initiate controlled freezing from the outside inward, preventing the random crystallization that ruptures cell membranes in any other frozen tissue. Simultaneously, the liver converts glycogen to glucose at an emergency rate, flooding every cell with a cryoprotectant concentration 200 times the normal level, replacing intracellular water before it can freeze and destroy the cell from within. The spaces between cells freeze. The cells themselves do not. At scale, this mechanism is the functional solution to the central unsolved problem of human organ preservation for transplant. Current medical technology maintains organ viability for hours before irreversible cellular damage begins. The wood frog maintains complete organismal viability for months, using proteins and glucose conversion pathways that natural selection refined over 100 million years. Medical researchers have been studying the wood frog's cryoprotection system since the 1980s. They have not replicated it in a mammalian system. The frog performs it twice a year in a pond. Tardigrade. In 2007, the European Space Agency attached tardigrades to the outside of a spacecraft and exposed them to open space for 10 days vacuum. Cosmic radiation, full solar UV spectrum, temperature extremes. When the spacecraft returned, the majority of the tardigrades were alive. A significant portion had reproduced during exposure. No other animal has survived unprotected open space. The tardigrade did it and laid eggs. The cause is cryptobiosis, a survival state so extreme that calling it dormancy is technically inaccurate. Under conditions that threaten viability, tardigrades expel 97% of their body water, retract all limbs, reduce metabolism to 0.01% of measurable baseline, and form a protein shell called a ton. In this state, no metabolic process detectable by current instrumentation continues. The organism is not surviving hostile conditions. It has exited the category of living organism as defined by metabolic activity and is waning as inert biological material for conditions to permit re-entry. The scale of where tardigrades have been found maps precisely onto the scale of their tolerance. They are the most widely distributed animal on Earth, present in every habitat sampled for biological life, without exception. From deep ocean sediment to Himalayan glacier to Antarctic bedrock to the upper atmosphere, In 2019, a spacecraft carrying tardigrade samples crashed on the lunar surface. Scientists who analyzed the crash concluded the tardigrades likely survived the impact. There are almost certainly living tardigrades on the moon right now, in the wreckage of a failed Israeli lunar lander, waiting in cryptobiosis for conditions that will not come. No protocol currently governs what that means for planetary protection policy. The question has not been formally resolved by any space agency. Planarian Flatworm researchers at Tufts University trained Planarian flatworms to navigate a maze. Then they removed the worms' heads. They waited for the heads to fully regenerate approximately 2 weeks and tested the new organisms on the same maze. The regenerated flatworms navigated it correctly. Memory encoded in a brain that did not exist 3 weeks prior. Reconstructed in neurons grown from cells that had never functioned as neurons. From information stored in a body that no longer contained the structure the information was originally encoded in. The experiment has been replicated. No complete mechanistic explanation has been published. The visible result begins with a simpler fact. Cut a planarian in half and both halves become complete organisms. Cut it into 200 pieces and each piece regenerates a complete flatworm brain, nervous system, digestive tract, reproductive system from scratch in 2 weeks. The cause is a stem cell density with no equivalent in vertebrate biology. Between 20 and 30% of every planarian's body mass is composed of pluripotent neoblasts, cells capable of becoming any tissue type the organism requires. When damage occurs, neoblasts migrate to the site, reference a body plan template maintained somewhere in the organism's cellular architecture, and reconstruct exactly what is missing. The process is not generalized regrowth. It is directed, accurate reconstruction of specific structures in the correct proportions and positions. The scale of what the memory experiment implies has not been absorbed into mainstream neuroscience. Where memory is stored in a system that can be completely excised and fully regrown without information loss is a question Thomas Hunt Morgan first posed when he cut a planarian in half in 1901. It remains open. The flatworm has been providing the same answer, that memory survives complete destruction and reconstruction of its substrate for 125 years. Biology has not yet built the framework to explain it. Bdelloid Rotifer. Bdelloid rotifers have not had sex in approximately 80 million years. This is not a behavioral observation. It is a genomic one. No male bdelloid has ever been found in any specimen, in any sample, from any location, in any era of the fossil record where they are represented. The entire class reproduces exclusively through parthenogenesis, females producing females from unfertilized eggs for 80 million years of continuous existence across every aquatic habitat on Earth. Sexual reproduction exists in evolutionary biology, primarily to shuffle genetic material and prevent the accumulation of harmful mutations. The problem described as Muller's ratchet, without recombination, deleterious mutations should accumulate across generations until a clonal lineage collapses. Bdelloid rotifers have been accumulating mutations without recombination for 80 million years and are thriving. The standard model predicts they should be extinct. The cause is a DNA repair system that operates under desiccation. The conditions Bdelloid rotifers regularly experience when their aquatic habitats dry up. When a bdelloid desiccates, its DNA shatters into hundreds of fragments. When it rehydrates, a repair mechanism reconstructs the genome and, in doing so, incorporates foreign DNA from bacteria, fungi, algae, and other organisms present in the environment during the repair process. Approximately 10% of the bdelloid genome is derived from foreign organisms through this mechanism. They are not reproducing sexually. They are collecting genetic material from every organism in their environment every time they dry out and rehydrate. The scale of what this means for evolutionary biology is a direct challenge to the framework that makes sexual reproduction appear obligatory for long-term survival in complex environments. Bdelloid rotifers found a third option: horizontal gene acquisition through environmental DNA that no other animal is documented using as a primary adaptive mechanism. They have been doing it for 80 million years across every continent in habitats that dry completely, freeze solid, and return to viability in cycles. The animals have been surviving longer than most current animal phyla have existed. The textbook explanation for why sex is necessary does not account for them. They have not adjusted their behavior to account for the textbook. Hydra, the Hydra, does not age. 4-year controlled studies at the University of Kiel found 0 increase in mortality rate over time in tracked specimens, 0. The animals at the end of 4 years were biologically identical in viability to the animals on day 1. The researchers found no upper limit to the observation because there was no signal of decline to measure. The experiment did not conclude it was stopped. The visible cause is a stem cell maintenance system that replaces every cell in the organism on a continuous rolling basis. The Hydra you see today shares almost no cellular material with the Hydra that occupied the same body 2 weeks ago. The physical structure persists. The cellular substrate is entirely new. Aging, in every organism where it occurs, is the cumulative result of cellular errors that accumulate faster than repair mechanisms can address them. In the Hydra, the substrate in which errors would accumulate is discarded and replaced before the accumulation becomes consequential. The organism does not repair aging. It structurally prevents it by never maintaining cells long enough for aging to occur in them. At scale, the Hydra is not rare, exotic, or confined to a laboratory. It lives in ponds and slow-moving streams on every inhabited continent. It is, by population and distribution, one of the most common freshwater invertebrates on Earth. Biological immortality, the property that makes the Hydra the subject of aging research across dozens of institutions, is present in an organism that lives in standing water in parks and drainage ditches in every city on Earth. The animal that does not age is not in a deep ocean trench or a specialized habitat accessible only to researchers. It is in the water humans drain, chlorinate, and pipe away from their buildings every day without knowing what they are removing. The most significant biological property documented in any living organism exists at the scale of a common weed. In the exact environments humans have been systematically eliminating since the beginning of urban development, and it was formally described in the scientific literature in 1998. If you made it this far, you already see what every animal on this list has in common, and it is not that they are hard to kill. It is that they solved problems biology was not supposed to permit solutions to. Aging, cellular destruction, extinction-level radiation, 80 million years without sex, death, twice a year, on a schedule. None of these are edge cases. They are organisms living in ponds, in soil, in the atmosphere above your city, doing things that the frameworks built to explain life do not fully account for. If you want more of this, the mechanisms underneath, the research that cannot yet explain what the animal already does, subscribe. And one more thing, the Bdelloid Rotifer has been collecting DNA from its environment for 80 million years. The tardigrade may be alive on the moon right now. The Hydra is in a pond near you. Which one should we have started with? Tell us below. There is no wrong answer, but some answers are more interesting than others.